Our first speaker for today is Claudio Babiloni, who's a professor, renowned full professor of physiology at Sapienza University of Rome and the director of the Laboratory of Neuroscience of Human Higher Functions. His studies concentrates on the brain rhythms that underlie awareness and cognitive abilities as people age and develop neurodegenerative disorders. To investigate irregularities in these rhythms in disorders like Alzheimer, Lewy body, and Parkinson's, researchers use EEG and neuroimaging methods. The PD Waves Consortium was established by Professor Claudio Babiloni and is an international cooperative project devoted to developing this field of study that he himself has pioneered. He has held prominent roles, including co-chair of the Special Interest Group in Advanced EEG and MEG techniques in clinical research for the International Federation of Clinical Neurophysiology and a member of the advisory board for the Alzheimer Association International Society. Professor Claudio Babiloni is motivated by an interest in the human mind and awareness to better the condition of those that have been afflicted by these terrible diseases. His efforts have influenced neuroscience and served as an example for those who would come after him in their quest to comprehend memory, consciousness, and human cognition. We have the privilege of benefiting from Professor Babiloni's knowledge and profound expertise that is truly remarkable, and I suppose at least as remarkable as the insights he will give us today. Professor Babiloni, thank you for being with us.
Thanks a lot for this nice introduction and for inviting me to this event. The title of my presentation today is Resting State EEG Rims Reflecting Vigilance, a Window of the Level of Consciousness. You see in the first slide my co-authors of this presentation. Human consciousness can be framed in two main dimensions: the level of consciousness or vigilance or wakefulness or arousal. And you can see that you can start from coma or general anesthesia, and then you can have sleep, drowsiness, and then wakefulness and full consciousness. The other dimension is the content of consciousness or vividness of contents of consciousness. And you see that you have a continuum in which you can have more and more vivid consciousness contents. And you can frame the mind states of consciousness into these two dimensions. And you, for example, see the continuum from deep sleep, light sleep, drowsiness, and conscious experience in relation to level of consciousness and vigilance and content of consciousness. One important point is the neural basis of these levels of consciousness and vigilance. And an important Russian neuroscientist in the last century introduced the theory of the working brain. And he conceptualized three main brain units. The first unit was involved in the regulation of a sleep-wakefulness cycle and the regulation of a vigilance consciousness level. And you see in gray the brainstem, the reticular ascending formation, and the interaction with the thalamus and cerebral cortex to modulate the vigilance and consciousness level. The second unit was involved in perception, memory, and emotional reactions, and was based on the posterior cerebral cortex in gray, you see, and the limbic system, amygdala, the hippocampus, and anterior cingulate, medial prefrontal cortex. The third unit was related to motivation, executive functions, planning of behavior, and was basically related to the frontal lobe and prefrontal lobe functions. So one point is how to study today the neural basis of vigilance, consciousness level with our brain techniques. And this is my proposal of 3 ways to analyze the neural basis of consciousness. The study of dynamics of brain activity by means of EEG or magnetoencephalography or functional magnetic resonance imaging. The investigation of the connectome, the structural connectivity in the brain, and investigate the causality of the role of different regions by brain stimulation, for example, transcranial magnetic stimulation. I will start with dynamics. The German psychiatrist Hans Berger was the first to record EEG activity, electroencephalographic activity from humans. And you see that we can do it with electrodes over the scalp and recording in different conditions of consciousness, vigilance level. You see that in the case of resting state, eyes closed condition, you can record an ample oscillations with dominant 8, the alpha rims, quiet vigilance, quiet level of consciousness. And if you are engaged in some mental operation, you see that there is the block of these alpha rims, the desynchronization of alpha rims. And you can see a substitution of higher frequency in the EEG from higher than 20 hertz, the beta and gamma rims. And you see that from the same electrode in the same person, if there is some drowsiness and then light sleep or deep sleep, you can see that you can observe dynamic changes in the amplitude and frequency of the EEG. So EEG seems to be nice to investigate the dynamics of cortical activity in relation to vigilance consciousness levels. And you see that Vincenzo Crunelli in his group of in Cardiff was able to demonstrate that with local field potential and in EEG in restricted brain regions, you can record in the wakefulness in cats and mice alpha rims in the sensory areas. And this is related to a similar oscillations of neural activity at the thalamic level. And you see that the up and down of oscillations of local field potentials are related to dynamics of spiking and silent periods in the single unit recording in the thalamus in relation to cerebral cortex. So you see that when you record EEG from the scalp, you are recording an oscillatory activity from the cerebral cortex, but in relation to thalamocortical interactions, but also in relation to ascending subcortical arousing systems using, for example, acetylcholine as a neurotransmitter in yellow, or dopamine from dopaminergic nuclei in the brainstem, or serotonin in the reticular ascending formation using this neurotransmitter, and also noradrenaline, and so on. So you can put in relation changes in the dynamics of EEG activity with vigilance, consciousness level, and the thalamocortical interactions, and the neuromodulation of dopamine, acetylcholine, and noradrenaline ascending systems. In our approach, we record EEG from the scalp of the humans, and we select artifact-free EEG periods, and we use these EEG signals as an input for Illoreta freeware to estimate the cortical sources of EEG activity. And we average the cortical source solutions within large regions of interest. And you see here an example of these cortical sources of resting state, eyes closed, EEG runs. And you see that in the vertical axis, you have the source activation. In the horizontal axis, you have the frequency band of EEG from delta to gamma, with a special attention to alpha. And you see here the cortical regions of interest in the Illoreta space. In this experience, we wanted to demonstrate that changes in alpha rhythms in the resting state, eyes closed, are related to changes in the vigilance consciousness level. And we use one night of sleep deprivation to reduce the vigilance consciousness level. You see that the recording before the night of sleep deprivation are in blue, and after one night of sleep deprivation, we had resting state EEG recordings in the red, in the same individuals, healthy people. And you see that before the sleep deprivation, you have a dominant source activity in the alpha frequency, in the parietal, occipital, cortical regions of interest. And you have a big drop of alpha source activity after one night of sleep deprivation. Please remember that alpha RIMS means cortical idling, cortical relaxation, inhibition, ready to respond, but in a resting condition. So we moved to investigate the relation between alpha RIMS and the contents of consciousness. And the experiment was based on giving a cue visual stimulus, left or right, so fast to allow just that in 50% of cases, the person could say, okay, there is a cue stimulus in the screen, left or right. And then you see that we use a mask stimulus, and then go. The subject move in relation to the green go stimulus. And after the movement on the mice, we ask, did you see the cue stimulus before? And he could say yes or no, seen or not seen. And we divided the EEG data in the events seen and events not seen. And we estimated the cortical sources of alpha RIMS before the cue stimulus. And we compared the data. And you can see in blue the alpha source activity. And it's very clear in the occipital, parietal, and premotor areas dominant before the people were able to recognize the cue stimulus. So big alpha activity before the Q stimulus, put the vigilance level in the perfect condition to recognize the following visual spatial stimulus. And then we also analyze the part during the Q stimulus, immediately after the Q stimulus, with the reduction of alpha rims, desynchronization of alpha rims as a sign of cortical excitation. And we could demonstrate that during the Q stimulus, the reduction of alpha rims predicted the ability to recognize the visuospatial Q stimulus. And you see the hyper desynchronization reactivity of alpha in the occipital and parietal areas. Then we wanted to test if these parietal alpha rims are really causative of conscious recognition of the visual stimulus. And we use transcranial magnetic stimulation to interfere with the parietal cortical activity during the Q task I described. And so we targeted the posterior parietal cortex, in which we observed these reactive alpha rims during the Q task. And we used TMS to interfere during the observation of the task. And we use also sham, a fake stimulation in the frontal midline as a control. And these are the results. While the subject was performing the task, the Q task, we gave the TMS stimulation in the parietal cortex, in the left posterior parietal cortex, in another condition, the right posterior parietal cortex, and in another condition, a fake stimulation on the frontal midline. And you see that if you use the sham condition as a reference, you have the number of Q stimulus consciously detected. And if you compare this mean value with the value of conscious detection of a Q stimuli in the same task, but during the TMS parietal stimulation, you see that you have a small but significant decrease of conscious perception of the Q stimuli. This means that these posterior parietal areas are causative in the experience of conscious detection of visuospatial stimuli in our task. Another chapter was to investigate the underlying connectome, brain structure, integrity of brain structure in relation to the regulation of vigilance consciousness level. And so we used some diseases in which we know the brain lesions. And we investigated the EEG resting state activity to map the vigilant consciousness level of these patients. And in our first study involved patients with persistent vegetative state. They are persons with a brain lesion supratentorial. This is the tentorium in red, and they have lesions upper the tentorium. They have a normal wake-sleep cycle, so they sleep and then awake during the day, but they are not reactive to environmental stimuli. They do not reply to questions of the experimenters and the physicians. family people. And we compared the resting state EEG runs in control healthy subjects in the upper row, and patients with the persistent vegetative state recovering after 3 months, and patients with persistent vegetative state not recovering consciousness after 3 months. At the time of EEG recordings, both groups of patients were in the same clinical condition of a persistent vegetative state. You see in the map, nose up, in the map, that in healthy controls, the dominant source activity is in the alpha frequency. Red means high cortical source activity, and especially in the posterior and the midline cortical areas. If you compare the patients with persistent vegetative state and not recovering after 3 months, you see that you have a dramatic reduction of alpha source activity, but you can still see some alpha source activity in red. And if you consider the patients not recovering after 3 months, you see that you really have a dramatic disruption of alpha source activity. So you can put in relation the alpha source activity with the level of vigilance and consciousness, and see that even if they have the same diagnosis, the same clinical evaluation, they have a difference as a group in the vigilance consciousness level as reflected by alpha source activity. Another class of patients of interest were Alzheimer's disease patients. Alzheimer's disease is a neurodegenerative disease related to the accumulation of toxin proteins, alpha, beta amyloid in blue, and phospho-tau in orange. And you see that in the early stages of this disease, belonging to severe cognitive deficits and disabilities of daily living at the end of the disease course, dementia. You see that in the preclinical stages, when you have a good cognitive status, you can detect some pathological abnormal accumulation of phospho-tau in the brain in orange. And then when you have the first objective cognitive deficits, you see that you have also a very clear accumulation of amyloid in the brain in blue. And when you have a severe cognitive deficit, dementia, and disabilities, you see that a big accumulation of amyloidosis and tauopathy. And so we investigated and compared this EEG sources of resting state EEG as closed in 2 groups of patients with mild cognitive impairment. In red, patients with the neurobiological diagnosis of Alzheimer's disease by means of the measures of amyloidosis and tauopathy in the cerebrospinal fluid. You see the lumbar puncture and so analysis of the cerebrospinal fluid. So in red, you have patients with Alzheimer's disease and mild cognitive impairment. And in blue, patients with mild cognitive impairment, the same age, gender, and the same cognitive profile, but not due to Alzheimer's disease based on cerebrospinal fluid. And you see that at the group level, you have a drop of alpha source activity in the occipital and parietal cortical regions of interest in the ilioreta space in Alzheimer's disease MCI. So you can see that even if the same diagnosis of cognitive impairment, there is a difference in the consciousness vigilance level as reflected by alpha source activity. We know that in Alzheimer's disease, we have a cholinergic impairment, a big cholinergic impairment, and we give cholinergic drugs to Alzheimer's disease patients in order to improve the cognitive deficits. And you see that today we can detect the connectivity, the connectome from basal forebrain to cerebral cortex related to cholinergic tracts, cholinergic bundles. And you see in colors different cholinergic bundles from basal forebrain to cerebral cortex in as templates in magnetic resonance imaging, thanks to Mesulam's group in Boston, in Chicago, sorry. And we compared normal elderly subjects with a group of mild cognitive impairment due to Alzheimer's disease, but with less cholinergic lesions. With a group of Alzheimer's disease MCI patients with significant cholinergic lesions. And you see that in the normal elderly subject, you have a nice alpha source activity in the posterior areas in red. You have a reduction in mild cognitive impairment due to Alzheimer's disease with less cholinergic lesions, and a dramatic drop of Alzheimer's disease in Alzheimer's disease MCI with substantial cholinergic lesions. So we put in relation cholinergic systems integrity with the generation of alpha rhythms and a regulation of vigilance consciousness level. Other patients of interest are those with Parkinson's disease and Lewy body disease, two variants of Lewy body neuropathology in the brain. And Parkinson's disease are affected by motor deficits, tremor, rigidity, small movements, and so on. But they have also some cognitive deficits. And you can see here the profile of cognitive deficits in Lewy body dementia, Parkinson, and Alzheimer. And you see that in these cognitive functions, we have some deficits, a bit specific for the disease, but a large overlapping. My point is that people are interested in the cognitive functions in these patients, but not neurophysiological measures of vigilance regulation, with the exception of Lewy body dementia, in which EEG recordings are supportive for the assessment, but no in Alzheimer's or in Parkinson. In reality, there are a very big difference in the alpha rims and in the delta rims in the resting state eyes closed in the three kinds of diseases. And this means that there is a difference in the regulation of vigilance and consciousness levels. You see in the graph in blue, normal elderly subjects with a big alpha source activity in parietal occipital areas during resting state, eyes closed, and a drop in green in patients with Parkinson's disease and dementia, even more in Lewy body dementia in violet, and a dramatic drop in Alzheimer's disease dementia. So the three diseases belong to dementia, but there is a difference in the vigilance consciousness level are reflected by alpha source activity. If you consider patients in the prodromal stage of dementia, mild cognitive impairment, no disability in the daily life, substantial disabilities in the daily living, you have the same pattern. Big alpha in healthy subjects, drop in Parkinson's disease with mild cognitive impairment, and even more in Alzheimer's disease with mild cognitive impairment. So the same kind of vigilance, consciousness, dysregulations can be seen even in the mild cognitive impairment condition, with differences between Parkinson and Alzheimer's. These patients are interested for visual hallucinations, Parkinson's disease with dementia and the Lewy body dementia. They have visual hallucinations, and in Lewy body dementia, this is very important in the assessment because they are frequent, recurrent, fluctuating, intense, and disruptive, even more than in Parkinson's disease dementia. And so when we compare the Parkinson's disease with visual hallucinations plus and no visual hallucination minus, red and green, we could observe that in relation to normal elderly subjects in blue, we have differences in alpha source activity in relation to visual hallucinations. So the consciousness vigilance level is effective in the generating a background for visual hallucinations in Parkinson's disease with cognitive deficits. And in another experiment, we used dopamine administration in Parkinson's disease patients. And we recorded before and after the administration of dopamine. And we observed that before blue and after administration of levodopa in red, we have changes in alpha and delta rhythms. So the vigilance consciousness level is related to dopaminergic and cholinergic neuromodulation. And when we consider the visual hallucinations plus and minus in dementia with Lewy body, and we compare the EEG, we did not see differences in alpha source activity, red and blue here. They are reduced, but in both groups, with visual hallucination or without. And the difference were in delta, in parietal delta. And when you have more abnormal delta in red, in patients with visual hallucinations, you have a more disconnection of the cerebral cortex than with alpha abnormalities. And this can be related to the fact that dementia with Lewy body have more visual hallucination than Parkinson's disease with dementia. So I can conclude that cortical EEG alpha rhythms underpin the level of vigilance and consciousness in humans and can be used to measure with neurophysiological techniques this state of the mind. They are poor in sleep deprivation. They are poor in patients with persistent vegetative state. They are poor in Alzheimer's and Lewy body diseases in relation to neuropathology and cholinergic deficits. And they are modulated in Parkinson's disease in relation to dopamine modulation. Thanks a lot for your attention. These are my co-authors, and these are my European grants.
Guys. Professor Babiloni, Thank you for the excellent talk. Now we give the opportunity to the public to ask one question if they are so inclined. Anyone? Okay, so this time I will do it. Do you think, Professor Babiloni, that in a meaningful way, AI can help us automatize detection and probably treatment of neurodegenerative disorders of any kind? Can we use such technologies to automatize the process of diagnosis?
Yes, this is something that we have to do, we are doing, because of course it's the only way to put together different parameters measuring the integrity of the brain in these patients and different way of neurodegeneration in different diseases, and functional measures, for example, PET with metabolism in the brain, or EEG in the dynamics of EEG rings, and also the cognitive assessment. If you want to establish the links between these dimensions, you need, of course, nonlinear classifiers such as tools of artificial intelligence, machine learning, supervector machines, artificial neural networks, deep neural networks, and so on. So I perfectly agree that the artificial intelligence will allow a step forward. However, it's important that we use the explainable artificial intelligence in order to always control which variables are effective in the discriminations of the patients with different diseases or the prediction of the disease progression and so on. We have to control that substantial and relevant parameters are playing a role in this classification and prediction to avoid to have non-replicable results.